Yes, me too. I'm very excited, and this is why I'm really happy that you are with us today. And with that, I'm going to, with no further, very minimal introduction now that we had this icebreaker kick off the first talk. So Marta Stefanska, so you are a PhD student at the Biozentrum in Basel with Professor Sebastian Hiller, and you are going to talk about Photo-CIDNP for diagnostics. So the floor is yours. You can share your screen, and looking forward to your talk.
Yes, just 1 second. I think that you should see it already. Okay, and okay, can you see it?
Yes, it's perfect.
Okay, so thank you very much again for kind introduction and for opportunity to speak here. So during the last webinar, we could see that Photo-CIDNP can be actually very successful in metabolomics. So increase intensity of some specific signals of components of complex biofluids such as urine or serum. And today in my presentation, I wanted to talk about a potential of Photo-CIDNP in target diagnostic. So I'm going to talk about finding new targets with machine learning, about matrix interference effect that unfortunately affects our Photo-CIDNP enhancement. And finally, show you some strategies how we can successfully quantify our signals in these very complex matrices that interferes with our signal. So I think that in this group, I don't really have to convince anybody that NMR is a really great technique that has a lot of application and gives you quantitative results. It's very stable, and with NMR, we basically can identify all the components of our sample by looking at specific fingerprints. However, its application in diagnostic is quite limited, and this is because NMR faces a lot of limitations. Limitations that actually can be overcome with high polarization, such as photochemically induced dynamic nuclear polarization I'm going to tell you about. So the first thing that NMR is notorious of is low sensitivity. Meaning that usually we have to have a highly concentrated sample or acquire a lot of scans in order to get a nice NMR spectrum and improve our signal-to-noise ratio. However, in biological samples, it's not really something feasible because usually like all the biological compounds are at very low concentration, and we cannot really prolong our measurement because this extended measurement could lead to sample aging, which alter the composition of our sample, and then we cannot really relatively quantify our compounds. So actually we can overcome this issue with Photo-CIDNP. By increasing intensity of some specific signals. Another problem with NMR is that usually spectra, especially coming from these complex biofluids, are very crowded, and we have a lot of overlapping peaks, and then extraction and quantification of the signals that we are interested in can be quite challenging. Of course, there are tools to do this, but it's still quite challenging. And during the last seminar, when Dr. Lars Kuhn was showing the metabolomic approach of Photo-CIDNP, we could see that actually we can use some special pulse sequences to get rid of all the thermal signals of the Boltzmann distribution and keeps only signals related to Photo-CIDNP. And because of this, we can significantly simplify our spectra. And the last thing that is a really big problem with NMR is that this technique is extremely expensive and requires demanding maintenance. And therefore, it cannot be really applied in hospitals or diagnostic centers. But fortunately, with Photo-CIDNP, we can get rid of these high field spectrometers and switch to low field spectrometers, to benchtop spectrometers, and that are relatively cheap, comparable with the state-of-the-art instrument used in diagnostic nowadays. They are very simple to use. And basically there is no maintenance. So I think that I show you that there is a lot of underlying potential in Photo-CIDNP as a diagnostic method. However, before we will be able to introduce this technique to the market as a diagnostic tool, we have to think about a few stuff that have to be investigated. And these are the same are my project objective, the stuff I'm going to tell you about during my talk. So the first problem is the space of potential Photo-CIDNP active targets. And this is a very important issue. And actually, this is not something trivial because not all the molecules are Photo-CIDNP active. And if they are, there is still huge variety in their performance, meaning that, for example, when we take the same scaffold and we just change the type of substituent at the same position, we can see that we can have completely different enhancement for our protons in the spectra. The same is happening when we change the position of the same substituent in our scaffold. We also have completely different enhancement factors and enhancement pattern. And of course, this is something that has to be resolved. Another problem is matrix effect, because so far it was shown in the literature that we can successfully quantify signals coming from photokidnap active targets in complex matrices, such as urine or serum or even E. coli extract, but we don't really know how quantitatively this matrix affects our photokidnap enhancement, if there is like any drop of photokidnap enhancement or not. So this is another issue. And finally, we have to think about strategies that has to be used to successfully quantify our signals in biofluids with all this matrix effect and the enhancement belonging to specific signals. So the first thing that we have to consider is finding this new potential photokidnap active target. And as I told you, this is not trivial because photokidnap depends on this very specific interaction between the dye and the molecule of interest. So far, to identify new photokidnap active targets, people usually do experimental screening, which is of course a great effort and brings a lot to the field, but it requires access to these big compound libraries and to high throughput setup to automate all the measurements. So this is not really feasible for all the labs. That's why I started working on developing in silico pre-screening methods that will be based on machine learning and enable fast identification of photokidnap targets that could accelerate the progress in the field. And then these targets could be used to my diagnostic tests. So the first thing that we have to start from is theoretical data, something that our model will be using to predict the photokidnap enhancement. So instead of acquiring directly experimental data, we want to predict this based on theoretical data. And of course, to be able to do this, we have first collect some experimental data and theoretical data as a training set for our machine learning models. And then we'll be predicting photokidnap enhancement for the test set not used in the training set. So the first thing that I had to do, I had to collect a lot of experimental and theoretical data. So I just measured a bunch of photokidnap spectra for molecules that were based on indole and phenol core. And I was interested in this type of scaffolds because they are very common in biological related samples and in drugs and in all the important metabolites. And I estimated the photokidnap enhancement of specific sites by signal-to-noise enhancement. And I also work with fluorescein as a dye because it's very resistant to photobleaching. So I could measure, for example, 16 scans. So this was the set of my experimental data. And now for theoretical data, I perform DFT calculation that are very easy to do even on standard computer without access to any cluster of molecular properties that are actually important for photokidnap. And I use both of this set of data, experimental and theoretical one, as input for my machine learning models to train and then to be able to predict photokidnap enhancement based on theoretical input. So now we have to consider which properties are actually important for photokidnap. So when we look at the general scheme of photochemically induced dynamic nuclear polarization, we can actually identify some steps that are very important for photokidnap to exist. So the first thing is that we have to have stabilizing interaction between the target and the dye, between the target molecule and the dye. And it was already shown in the literature that actually this can be correlated to the hydrophobic properties of the molecules. And this is because hydrophobicity of the molecules can be connected to aromaticity, which is responsible for pi-pi stacking between the dye and the target molecule. So they have, so they can stabilize the interaction. And because of this, we can go to another step. So the formation of spin-correlated radical pairs by electron transfer process. And here I included a bunch of parameters related to electron donating properties because our target molecule is basically electron donor. That yes.
We try to connect using. If they are busy, please leave a message. May I ask who's calling and why?
Sorry, again. Sorry, again, could you repeat?
Who was speaking? I think it was an automated stuff.
Okay,
Nina,
okay.
please just go ahead. Yeah.
Okay. I'm sorry.
Marta, sorry. I confused myself.
Okay, so yes, so for this step, I just included a bunch of properties related to electron transfer. So properties as isotope potential, nucleophilicity, HOMO energy level of the molecule of interest, and LUMO of the dye, and finally nucleophilic Fukui index, about which I'm going to tell you a little bit more. And finally, after this, we have a spin sorting mechanism, which is basically the core of all the hyperpolarization we get in Photo-CIDNP, and which is responsible of our hyperpolarization. And this is because we have a different triplet single interconversion for beta and alpha state, and this results in this population difference, which is higher than Boltzmann distribution. And in result, we have our increase in intensity. And because hyperfine interaction and delta G factors affect a triplet single interconversion, I use both these parameters in my models. And I also wanted to capture the interaction between them too. So I use modified equation for the geminate polarization that comprise the interaction between hyperfine interaction and delta G factors with a magnetic field. And this is actually something important because then the model has a potential to be extended to different magnetic fields, not only the one that I'm working on. So the first thing that I wanted to investigate, if we have like any direct quantitative interactions, if we have any direct interaction correlation between hyperfine interaction, between Photo-CIDNP enhancements and properties. If we can explain this in the quantitative way. So I tested different linear and quadratic fitting, and you can see the statistics shows us actually that we have some significance. But in the quantitative way, it's very difficult to directly explain our Photo-CIDNP enhancement. However, we can do this in a qualitative way when we think about nucleophilic Fukui index, which is basically a descriptor of how electron density change when we go from the neutral molecule to the cation. So important step of generation of radical parts because the electron has to go from our molecule of interest, from our target, to the dye. So here you can see that for all the molecules that I tested, I basically sorted all resonances according to increasing nucleophilic Fukui index with the bars height corresponding to the Photo-CIDNP enhancement. And usually the first one, which is the highest, usually the first one is the highest, meaning that nucleophilic Fukui index is actually a very good local indicator of the size with high Photo-CIDNP enhancement within the molecules. And the reason for this is that Fukui index comprises information about electron density change, about the electron donation, about the radical localization. And this is very important for hyperfine interactions because if the radicals is closer to nucleus, then we have stronger hyperfine interaction, which is here shown by this linear correlation between hyperfine interaction and nucleophilic Fukui index, which quite significant linear correlation. So from all the stuff that I show you so far, we cannot really rely directly on just one property to explain our Photo-CIDNP enhancement in the quantitative way. However, we can look in the reduced dimensional space to try to maybe see how the variance between all these properties look like, and then we can see that actually we have a very nice separation of region with high Photo-CIDNP enhancement, which is even better visible when we look at the 3D PCA with loadings that basically originate from all the features that we use. And thinking of this, we can actually consider this in this way, that, for example, some molecules can be very good in terms of stabilizing interaction between the dye and the molecule of interest, but actually fail in terms of any other factors like electron donation or, for example, hyperfine interaction and delta G. So this interplay, this matrix of different interaction between properties is actually something that we are interested in. And because for human, it's usually quite difficult to capture all this complicated interaction, I decided to evolve some machine learning models into this process. And here I'm going to show you just a very simple model based on logistic regression that's only purpose is to be able to, in semi-quantitative way, predict the class of our photokidnap enhancement. So the first thing that I did, I basically classified all my photokidnap enhancements in 3 different categories. Where you can see we have a quite huge imbalance between medium and high population. And this only shows you that actually this very high photokidnap enhancement above a factor of 90, it's not something very common. However, my models were able to first train on the test on the training set and then predict based on these theoretical properties that I calculated the photokidnap enhancement category for the tested molecules. And this is given by this confusion matrix. So basically, diagonal shows you the ratio of the correctly assigned category with high photokidnap enhancement predicted with 100% certainty. So this is exactly something that I wanted to achieve in this model. So be able to fish out the molecules, the size that have high photokidnap enhancement. That then later on I could use to my matrix to my photokidnap based diagnostic test. But before we go there, we have to think about like how everything else in our sample affects our photokidnap enhancement. So we have to consider how a matrix involves with this hyperpolarization that we have. So here I'm going to show you a very simple experiment when I basically measure the spectra for this 5-fluoro-2-methyl-indole, and we're going to focus on this resonance. So now when we at 600 megahertz measure a 100 micromolar sample of 5-fluoro-2-methyl-indole, you can see that in standard NMR, in the dark mode with no illumination, we have a very low signal-to-noise ratio. We probably would have to measure more scans. However, with exactly the same setup, but with a laser turn on, we can see that we have very high hyperpolarization, very high intensity of our signals. And now when we start adding some biofluids, in this case, I was adding urine, you can see that our signal drops. So actually, we have a very strong matrix effect. But even at 70% of urine, still our photokidnap enhancement is significantly higher than the signal that we recorded in the dark mode, so the one recorded with standard NMR. So by knowing this, we have to think about strategies that could be used to analyze our samples in biofluids. Because here I don't really have enough time to like show you all this interaction between matrix and different photokidnap active targets, but actually we cannot really rely on internal referencing. That's why I decided to work on the spiking method, which basically requires that first we measure unmodified sample with, of course, unknown concentration of our target compound. And then after this, we spike this sample with known amount of the target molecule. And from the difference in the kidnap enhancement, we can calculate the initial concentration. And for this, it's required that the correlation between integral, so our signal, and the concentration is linear. And this, you can see that for the region I was working on, actually this correlation is linear. And you can also notice that for samples obtained from 3 different patients, we have quite different slopes. And the reason for this is that this composition of these 3 different samples is quite different for 3 different patients. And then the matrices affect our photokidnap enhancement of the same molecules in a different way. And then based on this, we can back calculate the initial concentration that I tested, as I hear like 4 different concentrations for these 3 different patients with relatively small errors. And I verified this approach also for another molecule. And you can see that, again, we achieve relatively small errors. And another interesting thing that I observed is that actually photokidnap affects many resonances, but different matrices at high concentration definitely differ in terms of the slope of the calibration that we get. So this is something that you can see here. So at 5% of biofluid, in this case urine, we have relatively small standard deviation from the mean for these 3 different urines and 3 different resonances. However, when we increase the biofluid content, then the standard deviation for mean increases. Therefore, at 5% of biofluid in our sample, we can rely on universal calibration. So basically what I did here, I used samples coming from 3 different patients, and then I added only 5% of biofluid, and you can see that slopes are very similar in these 3 calibration curves, meaning that actually we can average them and then rely on sort of universal calibration to calculate our concentration without the necessity of using spiking. And then the technique that you want to use to quantify your signals, spiking or dilution method, is highly dependent on the quenching factor of the matrix effect and photokidnap enhancement of the target compound. And all the results that I showed you so far were presented for the high magnetic field. However, we can extend this to benchtop spectrometers. And this measurement in low field have this advantage that the photokidnap works actually better there. So here you have sample measured at 80 megahertz for 100 micromolar paracetamol. And to achieve this signal to noise ratio, we normally have to acquire 10,000 scans. So this is roughly like 20 hours. And with photokidnap, we can achieve the same signal to noise ratio with only 32 scans. So we can save a lot of time. And this is something very important for diagnosis. So actually here you can see the potential of photokidnap. And for a benchtop spectrometer, we can apply these 2 methods of quantification, so spiking and dilution. Here I just going to show you the result of the dilution method. So again, we have 3 different patients. The same target, you can see that the slopes are very similar, so we can average them and rely on universal calibration. And by this, we can think what is the future of photokidnap. So I think that is actually quite exciting in terms of diagnostic. Of course, there is a lot of things that we have to investigate first and better understand this matrix effect. It's something that I'm working on right now, because then when we will be able to understand really what is the main interference with our targets and with our dyes, we can think about some strategies of very simple sample preparation in order to eliminate this. And then we're not only getting higher enhancement, but we can also absolutely rely on universal calibration. And by this, I wanted to thank you very much for your kind attention, and many thanks to Professor Sebastian Hiller, who is a head of a structural biology group at Biozentrum, and who gave me this opportunity to work here on this project to learn a lot about hyperpolarization, NMR, and diagnostic application, and to Thomas Muttener, who is my direct supervisor and helped me a lot with technical setup and my project. If you have any questions, I will be happy to answer.
Great. Thanks a lot, Marta. This is an amazing talk. So please, everyone, ask questions in the chat. So you have the chat here. You can ask questions directly in there. Yeah, while people are asking questions, maybe I can open. I have one question for you, Marta. So first of all, it's very interesting to see this Fukui parameter that is actually quite good to mimic the hyperfine coupling constant, probably at much less computational time than if you would generate the radicals, right? My question is, have you tried to look back then into maybe some different molecules and scaffolds and indoles and phenols, like I'm thinking of purine or pyrimidine aromatics from DNA, right? Or from different aromatics that we can find in metabolomics. Have you investigated a bit different scaffolds, or have you stayed for now onto indole and phenols?
Not yet. This is something that will be in close future. Also implemented in my machine learning models. Yes.
All right. So, do we have any questions in the chat yet? Yes. So, Covadonga Lucas Torres is asking, thanks a lot, Marta, for the great presentation. The quantification method is impressive. How difficult is it to adjust target photosensitizer ratio when you do your spiking? And do you have to rely on one signal always?
So in terms of like adjusting the concentration of the dye and the target, I mean, like I use for the target, I use the the targets that are present normally in the physiological concentration. And for the dye, from my like pre-screening, I observed that usually concentration of fluorescein 50 or 100 micromolar works the best. But of course, it depends on the system that you are using. But this is not very difficult to be done. This is like just simple optimization. What I observe is like the matrix don't really affect how the ratio between the target and the dye is affected. So I mean, like you can like first do some very easy trials in buffer, estimate the concentration that you need, and then switch it to working on biological samples. And the second question, could you please repeat?
Yeah, do you have to rely on one signal always?
You don't have, of course, because like for some molecules you have many Photo-CIDNP active sites that have very nice Photo-CIDNP enhancement. But here for indels, I observed that usually I have like one resonance that have very high Photo-CIDNP enhancement. So of course I use it for my analysis. The same was for Sumatric then, but of course you can use as many signals as you want. Then you probably have more accurate results.